Hi to everybody. Thank you for joining us today, this evening, or from whatever time zone you happen to be joining us from. Welcome. I am Kathleen Duffy, Global Director of Life Science at Rheolution. So our purpose today with all of you, our community, is to share our knowledge of what we have learned through our collaboration with Nordmark. We have two presentations today, which should take about 30 to 40 minutes, depending on flow. And today we will follow up with the Q&A section. And speaking of Q&A, you'll see at the bottom of the page, there's a Q&A section. If you could please write your questions there specifically, and we will reserve the hands up section at the very end once we've sort of gone through the Q&A. So today we have two great speakers, Katerina Kemkens with Nordmark, as well as Dimitra Kamasau with Rheolution. So our first presentation will begin with Katerina, who will do a deep dive on critical components of effective cell isolation methods and tissue dissociation techniques, as well as look at enzymatic degradation characterization. So looking at common techniques to characterize the enzymatic degradation of tissues and or biomaterials. And then what will follow is the presentation with Dimitra with Rheolution, who will look at some of the new methods that we talked about to optimize your cell isolation protocols, as well as facilitate your workflow and make it more efficient, reliable, and cost effective. And what we hope that you will gain from this presentation is particular insights from these experts in the field who have hands-on experience in refining these cell isolation and tissue degradation methods, as well as acquire these practical tips and knowledge to enhance your particular techniques in the lab and provide you with even better results and ultimately elevate your cell isolation protocols. At the end of these presentations, we will have emails where you can reach us, as well as you'll be able to connect with us on LinkedIn should you have additional questions after these presentations. And with all of that, Katerina, please begin.
Hello. So let me just share my screen. Can everybody see the screen?
Yes.
Okay, wonderful. So welcome to our webinar. My name is Katerina Kemkens. I'm a product manager at Nordmark Biochemicals, and today with me is Karen Hinson-Reen, who is our representative in the US, and she might join us later on to answer particular questions you may have. So I'm going to start off today and give you an introduction on Nordmark and Nordmark Biochemicals, as well as collagen and its characteristics, and on collagenases and how they work. I will also show you our portfolio and explain to you why it is so important to have different collagenases with different properties. So as Kathleen already told you, whenever you have a question, please write them in the question and answer field, and we will come back to them at the end in the discussion part. So briefly about Nordmark Pharma, it was founded in 1927 with the headquarters being near Hamburg, and by now we have way over 600 employees. Our products are manufactured in a pharmaceutical plant, and we also do have DMFs filed with the FDA for our GMP collagenase products. We do have a dedicated production facility for our collagenases, so there is no risk with any cross-contamination. And therefore, we do offer an excellent quality and safety of the enzymes with also a high lot-to-lot consistency. And since we do have everything under one roof, we are proud to offer expert regulatory and technical support for our products. So all in all, you can say Nordmark combines manufacturing excellence with commercialization know-how. Nordmark Biochemicals itself is a business unit of Nordmark Pharma, and we are focused on collagenases and other proteases for tissue dissociation and cell isolation with a continuous product development. And I want to give you a brief overview on our product development. We do produce collagenase for cell isolation and use in cell therapy and regenerative medicine since 2002. And back then, we started off with collagenase MB4, 5, and 6, mainly for the use in stem cell isolation, and also collagenase MB1 premium grade and neutral protease, mainly for islet isolation. Both of these applications were our main applications back then and are still two of the most important ones. In 2005, we expanded the portfolio to also adapt to the regulatory requirements and therefore implemented our GMP grade version of collagenase MB1 and neutral protease. And since 2009, we also have the additional drug master files available for these GMP grade products to reduce the hurdles for our customers. 2015, we expanded our portfolio with the animal-free version to again make it easier and simplify the regulatory process, as there is no possible cross-contamination with animal-derived material. And in 2020, we added the collagenase MB70 gentle grade to meet the needs of our customers, and I will explain a little bit about this later on. So we're selling collagenase. So what is collagenase? Collagenase is an enzyme which can cleave or digest collagen. So it's produced by the bacterium Clostridium histolyticum and is an enzyme mixture. It is a composition of 4 enzymes mainly. So this will be collagenase class 1, class 2, neutral protease, and clostripane. And all of these enzymes target different parts of the collagen. So for example, the triple helix, the collagen fibers, and also other parts of the extracellular matrix. And they also do cut at different sides of the collagen protein. And therefore, you can say these 4 enzymes work together to dissociate the extracellular matrix. And because of these characteristics, collagenase is widely used for cell isolation from tissue, which is held together by collagen. So the question is, what's collagen? Collagen is the most dominant protein in the human body. It makes up to approximately 65% of the total protein in the body, and it is the main component of the connective tissue. So it holds the body together. And a lack of collagen may cause the body to literally fall apart. And there are a few main characteristics of collagen that may affect the efficiency of collagenase activity in your application. So speaking of this, that will be the collagen composition. So we're talking about the amount and the type of collagen. So far, there are 28 different types of collagen identified, and they differ by how the molecules are assembled, by added cell components, and also the location. But the main types of collagen are collagen 1 to 5. Another important point is the collagen cross-linking. So how is the collagen connected and to which extent? Also, the collagen density may affect your collagenase activity. So how is it packed and what is the size of the fibrils? And additionally, the orientation of the collagen in the tissue plays an important role. So all these factors may affect the experiment, and therefore a lot of questions should be asked before starting. For example, do I need lots of site activity due to the composition or the cross-linking? So we've learned that we have these 4 enzymes, which all have different cutting sites and targets. Do I maybe need a higher concentration due to the density? Do I need additional methods to crush or chop the tissue so my collagenase can get to the respective location where it could actually act? So this is, for example, important, especially if you have whole organ dissociation. So for visualization, what you can see here are different cell types in the native tissue. You can see the islet cells from the pancreas, osteoblasts, hepatocytes, and all of them cell types which are interesting for a lot of our customers. And what you can see is how different the tissue's extracellular matrix can be. So the collagen between the cells differs in the different organs, and the collagen differs even within an organ. And a more detailed example we found in the publication. And what you see here is that this group compared kidney and liver tissue regarding to the content and distribution of the different collagen types in fibronectin, which is also part of the extracellular matrix and interacts with the collagen. So the different types were stained, and as you can see, if you compare the upper and lower picture, the distribution within the tissue differs, as well as the amount of collagen. So in summary, there are a few main factors that affect the efficiency of the collagenase. We talked about the tissue structure, the collagen composition, arrangement of collagen, but also the chemical composition within the tissue, and also in the experimental design. So different collagenase grades are needed due to the different properties of tissues and cells and the different requirements of your application. And so far, the success of the degradation or cell isolation is mainly measured by cell count and viability. And an exact prediction is not always possible. So we do have a lot of experience regarding the different applications, but it's not always foreseeable how good a certain collagenase fits to a specific application. So every tissue requires a different collagenase for optimal digestion conditions. And therefore, we do offer several collagenase products with varying enzymatic activities for multiple applications. So Nordmark Biochemicals mainly has 2 groups of collagenase. On the one hand, crude or natural blend versus the purified ones. Whereas the natural blend is exactly what the name says. It's a natural and balanced composition of collagenolytic and site activity Clostridium produces. So again, remember the 4 main compounds of collagenase. And on the other hand, we have highly purified enzymes. So if you are on the left side of this picture, you see collagenase MB4 standard grade, which has a high proteolytic site activity compared to the collagenase activity. And moving to the right side of the picture, the collagenase activity rises in comparison to the site activity, meaning a higher purification grade of the product with NB7D gentle grade, which you heard before, is being partially purified. And moving more on the right side or the far right side, you find MB1 premium grade, having almost no site activity. And depending on the tissue and the cell type, a crude collagenase will work much better than a highly purified one, because these site activities are needed in most cases. And this is also the reason why, for example, a recombinant collagenase does not work so well very often. So you would at least need 4 enzymes to replace a crude collagenase from Clostridium. And there's no real benefit of a recombinant collagenase with respect to activity. So a highly purified collagenase may have the same performance, and the only difference is in production. And this is also the reason why we always recommend using our highly purified collagenases in combination with a neutral protease to have this needed site activity, but to be able to dose it individually depending on the application and the needs. So now we talked about the requirements of your application on your collagenase, but the choice of collagenase is not only based on the tissue requirements, but also, as mentioned before, there are also regulatory requirements of the authorities. And therefore, we offer a translational program. So you will find equivalent products produced under different standards, but with the same enzymatic properties. And this gives you the ability to go from bench to clinic and start with economical research grade, and then whenever you need it, switch to GMP grade, for example, for clinical studies. So, but you only need one protocol. And additionally, the animal-free enzymes we offer have the benefit that you don't have any cross-contamination with animal-derived components. So that really simplifies the regulatory process. So, and then to meet the requirements of the application and the tissue, as well as the requirements of the authorities, we offer a huge portfolio of different purification grades as well as production grades. And as we know, it is really hard to navigate through all the different options. You are always welcome to contact us, Nordmark product managers, and we are always happy to help you find the right collagenase for your application. We do have a lot of experience with the different applications and can give you some hints on what concentration might work best as a starting concentration, and also on your experimental conditions. So if you have any questions, you're welcome to contact us at collagenase@nordmark-biochemicals.com. But one question remains open. So how do you find the right collagenase for your application, the right concentration, the experimental condition, and how can you measure the tissue degradation efficiency? So one way I already mentioned would be the cell count and the cell viability in the end of the experiment. And one new way will now be presented by Dimitria, the Senior Application Scientist at our cooperation partner, Rheolution.
Thank you, Katarina. So I will share now my screen. So good afternoon, everyone. My name is Dimitria. I am a Senior Application Scientist at Rheolution. So now I will be moving to the second part of the presentation, where we will show you a project that we developed in collaboration with Nordmark about the characterization of tissues during the enzymatic degradation process. So first, I would like to thank you, Katarina, for the very nice introduction, and also all our colleagues that is involved in the organization of this webinar. And also, as Katarina and Kathleen mentioned in the beginning, if you have any questions, don't hesitate to add them in the Q&A section here at Zoom, because at the end of this part, we will read them. Okay, so I believe not all the audience is familiar with Rheolution. So we are a company that develops and produces modern laboratory and industrial instruments. We are located in Montreal, Canada, and we also have a commercial office in France. And we have some distributors spread around the world, as well as some our customers is spread around the world. And our first instrument is the Elastosense Bio, which is a mechanical tester that measures the viscoelastic properties of soft materials, such as hydrogels, polymers, native tissues, or actually everything that has a soft nature. Then we also have our new line of optical instruments with the 3BD, the Nefelo, and the Absorbi. These two first, they measure the turbidity of solution, which is related to the presence of particles in solution. And the Absorbi measures the absorbance of compounds present in the solution at specific wavelengths. And finally, we have our user-friendly software that is able to control all of our products through a tablet. I invite all of you to visit our website, Rheolution.com, if you want to have more information about these instruments. And also do not hesitate to contact us if you have any other questions after the webinar. So let's start with a brief introduction to the motivations of this product. As Katarina already mentioned in her part, one of the applications of their collagenase is to isolate cells or other components from native tissues, since they are able to break down the matrix of this tissue. And we also have enzymes being added into liquid media to mimic physiological fluids that can be added into biomaterials or medical devices in development to assess their performance in vitro in a condition that is more similar to in vivo. So how does this process work? We have here a very simple representation of an enzymatic degradation process. We have here a kidney tissue organ that represents the sample, the native tissue, or the biomaterial and medical device, that it's added in liquid media containing the enzymes. So this keeps mixing during a certain period of time under specific conditions, such as temperature, agitation, among others. And in the case of cell isolation procedures, this incubation may happen long enough to dissolve all the tissue for isolating better the cells. So some of the variables of this process includes the type of enzyme, as we saw in the part of Katarina. There are enzymes that are more specific to target tissues, to specific tissues. Also the dosage of the enzyme, as well as the concentration of other components in this digestion mixture. The digestion time, so the incubation time of the process, it's also a variable. And the digestion procedure, here it's a simple beaker, but there is also cases where bioreactors are applied. So we have a different experimental setup, and this can change the volume, the agitation pattern, the temperature, among others. So we can see here that there are many variables that can be optimized in this process. And then how someone know if the chosen variables were good or not. So in the case of cell isolation procedures, the professional may count the cells at the end of the process to see how many cells they could extract, and also they may perform a cell viability assay to see if those cells were alive or not. So let's say, for example, the person got a lot of dead cells. The person can optimize the degradation variables, can decrease, for example, the dosage of the enzyme or decrease the digestion time in order to see if this could improve the isolation yield. And then there is also another way to monitor the performance of the degradation process, which is characterizing the remaining tissue under the action of this degrading agent. So not just the cells at the end, you can also characterize the sample during this degradation process at different time points. And for that, some common characterization options would be measuring the weight of the sample. So weight loss is a common way to characterize degradation, or the mechanical properties as well during the different time points. And it is in this type of characterization method that we will focus on in our presentation. So just opening a parenthesis in this point, our mechanical tester, the Lasosans Bio, that I will be showing you soon, has been already used to characterize the degradation of chitosan hydrogels under the incubation of a lysozyme solution. So here the authors measured the weight and the mechanical properties of samples that were incubated with lysozyme solution in orange here, and of samples incubated with just PBS, which is here in turquoise. And what they could observe was that there was no change in weight during the 7 days of incubation, but there was changes in the mechanical properties from day 5 in between these two conditions. So the group demonstrated the highest sensitivity of using the mechanical properties to characterize degradation, especially at the beginning part of the degradation process. So then in this project, then in collaboration with Nordmark, we were interested in doing a proof of concept on the use of our mechanical instrument to measure the properties of native tissues, in this case, instead of hydrogels, during the incubation with collagenase. So just talking a bit about the instrument like that, the results will be easier to understand at the end. We have here the instrument and the software that was developed and is produced by Rheolution. We also have sample holders that are developed specifically for the instrument. We have different types. They are basically these cylindrical cups where the user add the sample inside, and then the holder is added inside the instrument. And at each measurement point, what the instrument does is that it applies a very gentle vibration. So the sample holder moves up and down. And then the way that the sample inside responds to this stimulation depends on the viscoelastic properties of the sample. So inside the instrument, we have a laser probe and we have a height probe that measures the movement of the sample from the bottom and from the top. And then these measurements are used in the algorithms of the instrument to calculate the viscoelastic properties. So we have as outputs the shear storage modulus, that is the G prime, the shear loss modulus, G double prime, and also other outputs such as tangent delta, the rate, and the height. So the instrument can measure the transition from liquid to gel when we are talking about hydrogels, for example, and that's why we see here an example of a curve, the G prime increasing over time when the sample is solidifying. But we can also measure a sample that it's already a soft solid, that it's already formed. And then in this case, we would have just flat lines representing the stable mechanical properties, which was the case of this project where we tested tissues that it's already formed. And one strength of our technology is that it's non-destructive. So we see here that there is nothing that touches the sample during the test. We have just ultrasound and a laser probe. So at the end of the test, you can have your sample. And this was relevant in this project because we were measuring the same sample multiple times during the degradation process. So this was more or less what we've done. We prepared the sample inside the holder. We measure it before the incubation with collagenase. Then we take it out from the instrument. We added collagenase for a certain period of time. And then after we tested again, and we did this multiple times. And other features of the instrument that it's relevant to this project is the fact that we can control the temperature of the chamber. So we've done the test at 37 degrees. And also we haven't used this feature in this project because we were doing short-term degradation, but when you are doing a long-term degradation, the instrument can be inserted into a biological flow hood. And then it has a mode that helps avoiding contamination of your sample during the test. So you can keep your sample sterile during your longitudinal study. So then I will move to the results. So the very first test that we've done, we tested kidney and liver in our macro volume sample holder that are those blue cups. So here we cut pieces, cylindrical pieces from these organs, and we introduced it in the holders. And we tested before any incubation with collagenase. And after we prepared the collagenase solution, we used NB4 and NB4G that were provided by Nordmark. And we used a concentration of 0.5 units per ml. This was a recommended starting concentration that we got also from Nordmark. So then we added this solution on the top of the samples. We let in the incubator for 30 minutes at 37 degrees. And then we removed the solution and we did a quick measurement in the instrument. So these were the results that we've obtained. Here I have relative G prime, so it's relative to T0 before the incubation with collagenase. We can see that liver and kidney with NB4, they reach a final G prime that it was 85% their initial G prime, so they had a decrease of 15%. And for the NB4G, we saw that the kidney was more affected, was more degraded under the action of the NB4G. And the liver stayed similar as the NB4. So these were preliminary tests that we've done, but we could already see the different performance here of the different enzymes according to the tissue type. And then for the next set of tests, we wanted to design a protocol in a way to accelerate this degradation process so that we decrease the sample size. So we used here our micro volume sample holder that allow us to add a smaller amount of sample. We've also cut the tissue into small pieces instead of having one block of tissue. Like that, we were expecting that the collagenase solution was going to penetrate better. And also because we are using the micro volume holder, this allowed the sample to be in contact with the solution from the top and also from the bottom of the sample. So we have collagenase on the top and bottom, and like this, we would also improve the penetration of the solution into the sample. For this set of tests, we used just kidney tissue. And we selected just the NB4G because it was the one that was more effective in kidney tissue. And here we increased the number of replicates to see also if we could see statistical differences. In this set of tests, we've tested 3 concentrations. So the 0.5 that we did before, and then we decrease roughly 3 times this value, and we increase 3 times this value. So 0.15 and 1.5 units per ml. So here we have the results at each concentration. We have here the absolute value. So we have the G prime over time. G prime, it relates to the stiffness of the sample. Over time, it was because we were testing the sample after 15 minutes, 30, 45, and 60 minutes of incubation. So overall, we could see that the shear storage modulus significantly decreased with the incubation time for all the concentrations. And if we see all of these results together in one graph, we can see here the relative G prime. So again, in relation to T0 for each one of the concentrations used. And we observed a significant difference in G prime between the 0.15 and the 1.5, so the extremes in concentration, after 30 minutes of incubation. And a significant difference in G prime after 45 minutes, also for the 0.5 and 1.5. And overall, we could see that by increasing the concentration of collagenase in solution, the degradation was happening faster. And also the extent, the amount of degradation was higher at 60 minutes of incubation. So we could see here that the sample at the end of the study, when we were using 0.15 units per ml, the sample had a G prime that was 50% the initial G prime. And then the highest concentration, so the 1.5, the final G prime was only 25% of the initial G prime. So we can see that there is a correlation, but we can also see that they are not strictly proportional, because here we are increasing 10%, more or less, in terms of concentration, and we're decreasing just 2, by a factor of 2, the degradation of the tissue. So as main conclusions, we've seen in this test that the type of collagenase can influence the degree to which different tissues undergo degradation, which is completely in line to what Katarina was mentioning in the beginning. In particular, here we saw that the NB4G had a stronger effect on kidney when compared to NB4. We also noticed that while an increase in enzyme concentration can lead to a higher extent of tissue degradation, the correlation between the two was not strictly proportional. And we can say that the mechanical properties of native tissues at different time points of the degradation process can guide the selection and the optimization of enzymes and extraction buffers, as well as other degradation parameters such as duration, volume, temperature, to achieve the desired degradation extent. So already in this short set of tests, we already use the results to optimize to select the enzyme, to optimize the concentration, to change the duration, and also to change the experimental setup. We used a smaller sample. We cut into small pieces instead of leaving a whole box. So already with these results, we could optimize the process to have a faster degradation. And the optimization of the degradation variables, in the case of cell isolation, the aim is to enhance cell extraction yields with the lowest cost and time. And for biomaterials and medical devices, these results can be used to tune the biomaterial itself to achieve better performances in a condition that is similar to the in vivo. So with that, I finalize the second part of our presentation. I would like to thank you very much for your attention. And we will pass to the Q&A section to see if you have any questions. But also, if you have any questions after this webinar, don't hesitate to contact us, Nordmark and Rheolution. You have here our names. You can find us on LinkedIn. And also, you have a contact email address to find us later. Thank you so much.
Wonderful. Excellent. So everyone, I don't know if you've had a chance to put your Q&A questions in. If you haven't, I can start with a question for Demetria. What would you say are the benefits with this new method specifically?
Yeah, so if we are thinking, we talked a lot about cell isolation. So if we think about this process, measuring the mechanical properties at different time points, it's a way to don't wait the process to finish to see if it has worked or not. It's a way that you can have data already during the process. So I think this is one benefit. And you can immediately act in your process to change it, to make it faster or make it slower. But of course, cell counting and cell viability at the end is also something necessary. So I see them more as complementary to each other.
Great. And I do have a question for Katarina. So you talked about how the chemical composition in an experiment may affect the efficiency of the collagenase, which which buffer should you recommend using for cell isolation?
So we actually do not recommend a specific buffer for our collagenase because we know from experience that several buffer types, including PBS, TRIS, HBSS, they all work for cell isolation with the collagenase. So, however, what you should consider is that collagenase activity depends on calcium. So therefore, we always recommend to add calcium to the buffer and at least 2 millimolar. And collagenases are inhibited by EDTA, so absolutely no calcium chelating agents should be in the buffer.
Okay. Thank you, Katerina. I have a question from Greg. Do you have functional data from your enzyme optimization testing? And if so, do you see the reduction in function if time is extended or enzyme concentration is too high?
So from this experiment?
I think maybe it's overall. I don't know if it's this particular particular one, but can you answer that question, Katerina?
So we do have
from the liver and kidney experiment you showed. Okay, yes, it's regarding this particular experiment. Yes.
So from this experiment, if we were running it for a longer time, we've seen that we didn't have much more decrease. So I would say yes for your first part, but this was just a small test, a small set of tests that we've done. We don't have extensive experience with collagenase. The enzyme concentration too high, with the concentration, the highest concentration I test was the 1.5, and we've seen that it degraded more. So I haven't tested more than that to see if it was giving the same results or not.
Great. I hope that answered your question, Greg. Yes, thank you. Okay, thank you, ladies. And then I have a question from Mohammed Tahir. What is the role of the cell proliferation? It has been checked. I don't know which one of you would be able to answer that question, or Karen. I don't know. Karen, since you are a guest on this panel, I don't know which one of you can answer this question.
I can say that in this particular project, we haven't seen the cells, so we were just characterizing the tissue during the degradation of the tissue. So in this one, we haven't checked the cells that were isolated.
Thank you. And let me just see, are there more questions? I think I might have one last question. What is the advantage of using the highly purified enzyme, and where are they used? That question is for you, Katerina.
Yes. So the advantage is that you can dose individually, and it is usually used for very sensitive cells. And one example would be the islet cells from the pancreas. And so and also for this specific application, it's also important because the concentration of both enzymes required for the isolation really depends also on the age of the donor or on the organ weight and condition. So depending on what condition should be treated and what condition is a pancreas in, different concentrations may be better to isolate these islet cells. Another area of application would also be the isolation of tumor cells, also very sensitive cells where especially the receptors are important and need to stay the integrity should stay as it is. An example would be the TILs, and yeah. So for these kind of cells, it will be beneficial to use a highly purified enzyme.
Great. Now let me just see in the Q&A. I don't have any more Q&A. Is there anything else, dear audience, that you would like to ask? You can ask it anonymously if you want. I think you have that choice. Otherwise, what I may ask, Karen, we didn't have a chance to hear from you, and I was just wondering if you had anything to add, or if there were other questions we should be asking that we haven't asked yet.
Well, I would mainly like to add that it's been a real pleasure working together with Rheolution on this project. Nordmark is always looking for ways to better support their customers, as is Rheolution, and this has been very exciting for us to find out more earlier on in the process what's happening instead of just cell count or cell viability. And so we really appreciate the cooperation and collaboration, and look forward to learning more about ways that we can work together, and maybe other people have other suggestions and ideas for us.
Yeah,
so
definitely. Yeah, definitely. Thank you, Karen. That's very generous, and it's reciprocal. So if there are no other questions, did we want to go on the hands up? Any raised hands that you need, that you want to use your voice in doing an exchange? If I don't see anything in the Q&A, I will assume not. I think we're good. Is there anything else that we wanted to add that we haven't had a chance to add throughout this presentation? Well, then on that note, I want to thank everyone who joined us, both earlier on this morning, as well as today at midday. So we really appreciate your participating, your questions, and we are available as well for further questions if others come about, as we mentioned, via email or LinkedIn. You'll be able to find us there. So thank you everyone for your contribution. We are open to discussing with you, and we look forward to the next webinar. Thank you, everyone.